Localization of proteins in the cell. Lecture 2, Protein Biosynthesis. Now, before we start on discussing protein biosynthesis, there are a couple of expressions or concepts that we need to agree on, and that's the proteome and proteomics. The vast majority of genes in the nuclear DNA encodes proteins. Now, the proteome comprises all the proteins in a given cell, tissue, or organism. Some of the proteins are tissue specific or are synthesized only under specific conditions. Proteomics comprise all the methods available for studying the proteome. To study the proteome, we first need to consider how many genes there are in a cell and how many proteins they encode. This table shows the size of the nuclear DNA and the number of protein-coding genes in various organisms. In Escherichia coli, that's the well-known bacterium, the size of the nuclear DNA is 4.6 megabase pairs. Now, this is an important prefix to understand or to know. Mega means million, 10 to the 6th. Now, those 4.6 megabase pairs encode about 4,600, include 4,600 protein-coding genes. In yeast, the size of the nuclear DNA is larger. It's 12.1 megabase pairs. And that DNA encodes 5,900 proteins. Then we come to Homo sapiens, ourselves. And the DNA is much bigger. It's 3,000 megabase pairs, which can also be written as 3 gigabase pairs. And that encodes 20,000, has 20,000 protein coding genes, which encode more than 30,000 proteins, which is due to alternative splicing of many genes. And then finally, we have plants. I always include plants because I work with plants myself. Arabidopsis thaliana, that's thale cress, 157 megabase pairs. So it's much smaller than the human DNA, but it still encodes has 27,600 protein coding genes, encoding more than 30,000 proteins. Again, some genes encode more than 2 proteins. So note that plants are actually just as complicated, or perhaps even more complicated than man. One small note here is that the 3,000 megabase pairs, the size of the human DNA, would in American or English probably said to be 3 billion base pairs. Now, I think one should avoid using billion because in many European countries, billion means 10 to the 12th, whereas a billion in the United States and the UK means 10 to the 9th. 1,000 million is much better. Right. Now, the proteins are encoded in the cell genome, the DNA. In the vast majority of that is found in the nucleus. However, mitochondria and chloroplasts contain a small amount of DNA, but that encodes so few proteins that we can ignore it in the remainder of these lectures. However, because we'll be using mitochondria as an example throughout these lectures, we need to consider the size of the mitochondrial proteome. Now, in yeast, the mitochondrial proteome, the number of proteins in yeast, different proteins expressed in yeast, is 1100 in the yeast mitochondria. In humans, it's 1200 to 1900. And again, plants are more complex. Arabidopsis thaliana mitochondria contain 2,000 to 3,000 different proteins. So, about 10 to 15% of all proteins in the proteome are found in the mitochondria. So, this slide shows protein biosynthesis. Basically, protein biosynthesis converts DNA to RNA, and then from RNA to proteins. As a general rule, one gene in the DNA encodes one protein. But as I've already mentioned, there's alternative splicing, which means that one gene can actually give rise to several different proteins. One gene is anywhere from 100 base pairs to 10 nucleotides to 10,000 nucleotides. There's a vast range. There are some very small proteins, and there's some very, very large proteins. Now, what happens is that DNA is transcribed into messenger RNA, which can be modified post-transcriptionally. Then that messenger RNA moves out into the cytosol, and then it is translated into proteins on ribosomes or by ribosomes. Now, the proteins of the messenger RNA consist of a chain of nucleotides that is translated to a chain of amino acids, a polypeptide chain. That is a protein. So, and I should add that the proteins can then be modified before they reach their final destination. Right. Now, the messenger RNA leaves the nucleus and comes out into the cytosol, where it binds to ribosomes. Now, those ribosomes can either be bound to the endoplasmic reticulum, making it the rough endoplasmic reticulum, or they can be floating around in the cytosol. But let's take a look at the bound ribosomes. Now, the bound ribosomes on the ER synthesize proteins that then go into the lumen of the ER. Vesicles with the newly synthesized proteins bud off from the ER and fuse with the Golgi, where the proteins are modified. For instance, sugar molecules can be added to some amino acids in the Golgi. And then, from the opposite side of the Golgi, new vesicles bud off and, for instance, fuse with the plasma membrane. Note that there's a sugar added in this example here, the purple bit, ends up on the outer surface of the plasma membrane. That's very important for immunological identity of cells in humans. Now that's bound ribosomes. So bound ribosomes on the rough ER are destined for ER. Of course, that's where the ribosomes are sitting. For the destined for the Golgi, for peroxisomes, for plasma membrane, as shown in the previous slide, and for the extracellular space. So the proteins in those vesicles that fuse with the plasma membrane can actually be released into the extracellular space. And those proteins in the vesicles can also fuse with the vacuole. On the other hand, proteins synthesized by free ribosomes in the cytosol are destined for the cytosol, not surprisingly, are destined for the nucleus, so the proteins go back into the nucleus, the mitochondria, and the plastids, the chloroplasts in plant cells. So to reach the right destination, each protein carries within it the information that allows it to be recognized by receptors at the various steps in the protein sorting process. Now, to appreciate the type of information carried by a given protein, we have to take a close look at protein structure and function in lectures 3 and 4, and then return to the question of protein sorting in lecture 5. So in lecture 2 on protein biosynthesis, we've seen that nuclear DNA encodes for more than 20,000 proteins in animal and plant cells. In fact, more than 30,000. Nuclear DNA is transcribed into messenger RNA, which is translated into proteins by ribosomes in the cytoplasm. The free ribosomes in the cytosol, that's the soluble phase of the part of the cytoplasm, translate proteins destined for the cytosol, the nucleus, the mitochondria, and in plants, the plastids. On the other hand, bound ribosomes on the ER translate proteins destined for other locations inside and outside the cell. And that concludes lecture number 2.